Observing microorganisms. By the end of this lecture, you'll be able to answer this question. Acid-fast staining of a patient's sputum is a rapid, reliable, and inexpensive method to diagnose tuberculosis. What color would bacterial cells appear if the patient has tuberculosis? This figure is a reminder of the comparative sizes of various types of cells, viruses, and cellular structures. You should be well aware of the sizes of prokaryotic and eukaryotic microbes in relation to each other. The compound light microscope uses light to view images of microbial specimens. The type of light microscope used in a teaching microbiology lab to view bacteria and larger eukaryotic microbes is known as a bright field microscope. Because viruses are so much smaller than microbes, they must be viewed with a more powerful microscope. The specimen is prepared for viewing and placed on a glass slide. The slide is then secured to the stage with stage clips. Some bright field microscopes are monocular, meaning they have one eyepiece, and some are binocular, like the one shown here, and has two eyepieces to view images. Eyepieces have an ocular lens that magnifies the image on the slide by 10 times. At the end of the revolving nose piece are objective lenses that further magnify the specimen image. The objective lenses vary by their magnification power. Common objectives found on a bright field microscope are 4x, 10x, 40x, and 100x. The total magnification of an image you view is a combination of the ocular lens and the objective lens and can be calculated with the equation shown here. Total magnification equals ocular times objective lens. In this example, the objective used to view a specimen is 40x. So the total magnification is 10x, the magnification of the ocular, times 40x, the magnification of the objective. 10x times 40x equals 400x. So the sample viewed using the 40x objective has been magnified 400 times. So if we look at the total magnification of a bright field microscope, that total magnification is 10x, the magnification of the ocular, times 100x, which is the magnification of the highest objective on a teaching microscope. So 10x times 100x is 1000x. So for a bright field microscope that has the highest magnifying objective of 100x, the total magnification or highest magnification of a bright field microscope would be 1000x. You should read section 2.3 in the online text and watch the video which describes the parts of the microscope and their functions. This figure shows the path of light through the microscope. Light from the illuminator or light source at the base of the microscope passes through the condenser lens, the specimen on the stage, enters the objective lens, and then detected by the ocular to view the image of the specimen. When an image is magnified, it is enlarged, but we also want to consider resolution of the microscope because we want a clear image. Every microscope has a designated resolution, which is the ability to distinguish 2 points as separate individual points. For example, if a microscope has a resolving power of 0.4 nanometers, then it can distinguish 2 points as finely focused and separate if they are 0.4 nanometers apart or more. If the points are closer together than 0.4 nanometers, they cannot be viewed as separate and will appear as one blurry point. As the light leaves the specimen, it is refracted by the air. We can increase the resolution of the bright field microscope to its maximum level by using the oil objective lens in combination with immersion oil. Using oil on this special objective lens decreases the amount of light that is refracted, increases the amount of light that passes through the objective, and therefore increases resolution or clarity of the image. Please watch this short video to see how oil decreases refraction and increases resolution. The electron microscope magnifies specimen images 1,000 times or more than a bright field microscope. These microscopes use electrons instead of light to increase magnification and resolution, allowing us to see internal cellular structures with a transmission electron microscope and cell surface structures using a scanning electron microscope that are impossible to see with bright field microscopes. These microscopes are powerful enough to see viruses. Samples viewed under a microscope must be prepared for viewing. Cells can be viewed in their natural state by placing a small drop of bacteria submerged in water or other liquid onto a glass slide and viewing them under the microscope. Cells viewed in a wet mount are virtually transparent and must be viewed with low light. Wet mount is a term used for cells that are not stained. Stains can be used to increase the contrast between the background and the cells. A simple stain is a dye that stains the cells directly. The positive charge of the dye in the stain is attracted to the negative charge of the cells. When the staining procedure is complete, the cells are the color of the stain and the background is white. Differential stains are used to separate bacteria into two large categories based on the stain. Differential stains use two simple stains of different colors. The cells are stained with the first simple stain. A decolorizer is added during the procedure and removes the stain from some of the cells. Then a second stain is applied at the end of the procedure to stain the decolorized cells a different color. Two differential stains commonly used in a microbiology lab are the gram stain and the acid-fast stain. A gram stain is the first stain used in the identification of unknown bacteria. Acid-fast stains are used to detect mycobacterium species like Mycobacterium tuberculosis, which is the organism that causes tuberculosis. The gram stain is one of the most performed stains in microbiology. It separates bacteria into two wide categories of gram-positive or gram-negative cells, which tell a lot about the structure of the bacterial cell envelope. Gram-positive bacteria appear purple after the staining procedure, and gram-negative bacteria appear pink or red. Gram-positive bacteria are generally more susceptible to antimicrobials and tend to be more easily killed by penicillin and detergents. Conversely, gram-negative bacteria tend to be more resistant to antimicrobials and detergents. You should know how the gram stain works and how to troubleshoot issues that may occur when performing this procedure. This procedure will be explained in a subsequent lecture. Pictured here is an acid-fast stain of Mycobacterium tuberculosis. Acid-fast bacteria have special components in their cell wall called mycolic acids that make them more difficult to stain and more difficult to destain. So, the stain and decolorizer contains acid and is used to stain and destain during this process. Acid-fast bacteria tend to grow very slowly, and the infections they cause should be treated quickly. Performing an acid-fast stain on patient samples suspected of having one of these bacterial infections helps doctors identify these organisms without waiting for slow cultures to grow, and patients can begin treatment sooner. Acid-fast cells are pink or red, and non-acid-fast cells appear blue at the end of the acid-fast staining process. Negative stains are used to stain cells that are surrounded by capsules, and they indirectly stain cells. The negatively charged colored stain is repelled by the negative charge of the cell. So in a negative stain, the background is colored and the capsule is unstained, leaving a halo appearance around the cells. Here are two different bacterial species that contain capsules. Capsules are associated with bacterial strains that cause infections. So identification of capsules are very important in diagnosing and treatment. Some organisms form protective structures called endospores. These structures can withstand very harsh conditions. The endospore stain stains endospores green with the malachite green dye, and all other cells are stained pink or red. The decolorizer for the endospore stain is simply water, and it easily removes the malachite green from the non-endospore, also called vegetative cells, that are subsequently stained with pink or red by safranin dye. This stain is important for identification of clinically significant organisms like bacillus species. For example, Bacillus anthracis. This organism causes anthrax, as well as Clostridium species that cause C. diff, which is an infection commonly acquired in hospitals. The last special stain is the flagellar stain. This stain specifically coats flagella with tannic acid or potassium alum and then stains the flagella with pyroxaline or basic fuchsin dye. Staining flagella helps microbiologists study motility and also helps in the identification and classification process of unknown organisms. This is the end of this lecture. Thank you for listening.